Module 7, Neural Networks Regulating Feeding. This video will provide a brief overview of the interacting networks that regulate feeding, with more details in the papers and videos to follow. So we return to the signaling network that was hypothesized, as we said earlier, in the 1950s. We know now that the signal protein is in fact the protein leptin, and that its receptor in the brain is the leptin receptor. And now we're going to extend this model to discuss the satiety factors and thermogenic factors that respond to leptin binding to the leptin receptor in order to regulate energy balance. This table provides an overview of the many neuropeptides and molecules that are important in regulating energy balance and satiety. And while they all play important roles, among the most critical would be neuropeptide Y, an agouti-related peptide on the orexigenic side, and cocaine and amphetamine-related transcript, or CART, and melanocortins derived from POMC on the anorexigenic side. In terms of the periphery, we know that leptin plays a critical role in signaling stored energy to the brain. And ghrelin is also an important molecule that's expressed in the gut, and it's one of the few gut peptides that actually promotes eating rather than stimulates satiety. The agouti mouse is one of the oldest mouse models of obesity. It develops morbid obesity post-weaning, which is correlated to the intensity of its yellow coat color. No defects in temperature regulation have been observed in these animals, although they develop hyperinsulinemia and hypercorticosteronism following the development of their obesity. They have increased linear growth, and they have severe hyperphagia, which is a primary behavioral phenotype leading to their severe obesity. It actually took a long time to figure out the genetic defect in the agouti mice. It was known that the agouti protein is normally expressed in skin, where it binds to the MC1R receptor, and the variable amount of agouti expressed is what determines the coat color from yellow to black. But it wasn't clear what the defect was that was causing obesity, until it was realized that a transposable element was basically disrupting the regulatory elements of agouti and causing it to be expressed in all cells, not just the skin. Within the brain, agouti competes for the binding of another molecule called alpha-MSH. Alpha-MSH binds to the MC4R receptor and induces satiety. It's a really important factor in controlling satiety, and it was actually the discovery of this defect that led us to our understanding of how the leptin system interacts with the melanocortin system to influence satiety and energy balance. Following the discovery of the genetic mechanism for agouti, a protein called agouti-related peptide was identified. It's a hypothalamic protein with high sequence homology to agouti, and it's elevated in obese and diabetic mice. Transgenic mice that are overexpressing AGRP ubiquitously are hyperphagic and obese. And AGRP, as it turns out, antagonistically binds to the melanocortin receptors 3 and 4, which are normally targeted by alpha-MSH. Neurons in the hypothalamus that express AGRP are essential for controlling energy homeostasis, and like NPY, AGRP is associated with extreme hyperphagia as well as compulsive behaviors. Pro-opiomelanocortin, or POMC, is a precursor protein that was cloned in 1972, and it's a great example of the fact that the original assumption of a one-to-one relationship between genes and proteins was absolutely incorrect. In the first stage of cleavage, adrenocorticotropic hormone and beta-lipotropin are produced along with an N-terminal glycopeptide. In the second stage of cleavage, both alpha melanocyte stimulating hormone, or alpha MSH, which is associated with satiety as we saw with the agouti mice, and beta endorphin, which is associated with pleasure reward and the runner's high, are produced. In 1999, a knockout model of POMC was created, producing yet another model of obesity. It had defects in adrenal development, and it had altered pigmentation similar to the agouti mice. One of the most powerful stimulators of eating behavior yet identified is called neuropeptide Y. When neuropeptide Y is infused into the brains of rats and mice, they continue to eat even after the stomach has become engorged with food. It's also a regulator of resting metabolic rate, and it's a powerful pressor agent acting to influence vasoconstriction and blood pressure. It's an important regulator of central endocrine function, and as it turns out, it's a really important regulator of the downstream effects of leptin. NPY and leptin receptor actually co-localize to the same neurons within the arcuate nucleus and within the median eminence of the hypothalamus. Infusion of leptin results in immediate downregulation of NPY expression and decreases in circulating NPY levels. As you can see in this figure to the right, leptin is being secreted from adipose, reaches the neurons in the brain, binds to its receptor, and then it has an inhibitory action on NPY. Infusion of NPY, alternatively, can also alter leptin levels. The surprise, given the strong phenotypes of NPY, is that the knockouts of NPY have no obvious phenotype. Cocaine and amphetamine-related transcript has actually been shown to be regulated by cocaine or amphetamine injection, yet it plays an important role in the brain, as it turns out, because it's highly expressed in the hypothalamus, and it plays a role in regulating satiety signaling. ICV administration inhibits feeding and inhibits NPY action, so it actually plays an antagonistic role to the action of NPY. It's reduced in OB mice, and it's restored by leptin administration. It's also co-expressed with POMC. Orexin is also called hypocretin, is expressed in the brain and has been shown to stimulate food consumption in a dose-dependent manner. The mRNA of orexin is upregulated by fasting, and it's co-localized with NPY, AGRP, and POMC. It may also be involved in drinking behavior, neuroendocrine function, and sleep-wake cycles. And in fact, the reason why it's called hypocretin is because it was actually first identified in a patient sample of narcolepsy. While we covered quite a lot of neuropeptides, we haven't nearly covered them all. But this beautiful picture shows how these neurons in the brain that express these various neuropeptides that regulate feeding behavior are highly intercalated among each other. And here you see this in this little cartoon to the left, how axons from the arcuate nucleus that are expressing NPY and POMC, so both orexigenic and anorexigenic, are now connecting and interacting with other downstream neurons and cells that express and amplify and continue those signals of either stimulating or inhibiting feeding. It's interesting to note that all of the single gene mutations that are known for obesity in humans are actually encoded in these satiety signaling pathways. And in fact, every single one of these mutations has associated with it hyperphagia, a behavioral phenotype as opposed to a physical phenotype. So clearly these pathways are very important for physiology and for homeostasis. So the combination of these many molecules interact and work together to maintain energy homeostasis. For example, when leptin levels fall due to dieting or due to starvation, the body responds by increasing levels of the orexigenic molecules that will stimulate eating behavior and decreasing levels of the anorexigenic molecules to release that inhibition of feeding behavior. As a consequence, raising appetite and decreasing energy expenditure, as well as other neuroendocrine factors. When adipose mass rises due to overeating or other means, leptin levels rise, and subsequently the orexigenic molecules fall and the anorexigenic molecules rise themselves, therefore having a double inhibition on feeding, which reduces appetite and increases energy expenditure. It's truly an amazing and highly interactive system, and we've only covered a small part of it in this little introductory lecture.